This chapter really covers how energy is going to be used in the cell. We're going to talk about specific definitions of energy, potential versus kinetic energy. We'll get into the two laws of thermodynamics. You'll need to know those definitions very well. We'll talk about the breakdown of the energy equation, which includes enthalpy, entropy, Gibbs free energy, and temperature. We won't solve any equations, but you definitely will need to know those terms, what they mean, how it plays into the changes from potential to kinetic energy, and the types of chemical reactions associated with them. We'll talk about the difference between spontaneous and non-spontaneous. Those are very specific and definitions that you'll need to know for this chapter and for the upcoming chapters with photosynthesis and cellular respiration. We'll talk about the differences between exergonic and endergonic. Those are how the energy is going to flow either into the system or out of the system. You'll need to be able to associate all of these things with potential versus kinetic energy. And so I'll show you a breakdown on how ultimately you'll have your notes set up so that you can see both sides of the coin. So conversely, what's going to be included in a chemical equation that includes spontaneous versus non-spontaneous, or exergonic versus endergonic, and so forth. So we'll get into that a little later. And then we'll talk about ATP hydrolysis. This is a specific chemical reaction in which energy is going to be released. This is going to be very important for the next two chapters as we talk about how the cells are going to either make ATP or break it down in order to use that energy to do some work inside the cell. And then at the very end, we'll talk about how enzymes are playing a role in this flow of energy, and then a specific type of catalyst called a ribozyme. You'll need to know that definition, the difference between ribozymes and enzymes as we move forward. Okay, so energy is the ability to promote change or do some work. There are two forms of energy. That energy can either be in the form of kinetic, which is associated with movement, or it can be in the form of potential or stored energy. And generally, when we're talking about kinetic energy, we're talking about what happens when energy gets released from chemical bonds. And with potential energy, we're talking about how energy is stored in those chemical bonds. Remember this definition about where the energy is stored in chemicals, and those the energy is going to be stored in the bonds. And so we've talked about building, we've talked about breaking, hydrolysis versus dehydration synthesis. Those things are important here again as we're talking about energy being stored versus energy in motion. And so over the next two chapters in photosynthesis and cellular respiration, we're going to be talking about how the cell is going to be storing that energy. For example, in photosynthesis, when the plant cells are going to convert light energy and store it into the bonds of glucose, for example. And then we'll talk about how that energy is going to be released in cellular respiration. As you eat food, your body then takes that food, breaks it down into various components, and by breaking down the food, the bonds of those food molecules are broken, and in the process, that energy is captured and converted into ATP. So that's this picture here with the roller coaster going up the hill. We'll watch the roller coaster go up the hill. You can see potential energy is increasing. And then as the roller coaster goes down the hill, kinetic energy begins to increase. So there, again, two sides of the coin: potential stored energy versus kinetic, which is energy in motion. This molecule that is here to the right is ATP, and you can see the pink arrows are pointing to those bonds in between the phosphorus. Those phosphate bonds are high energy bonds of ATP. You can see we have a phosphate, a sugar, and a base. And so of the phosphate, we have three phosphate molecules, and those three phosphate molecules are where the energy is going to be stored. So we'll see in the next, over the next two chapters, where in photosynthesis, The energy is going to be stored in those phosphate bonds during photosynthesis, and then in cellular respiration, we'll see how those phosphate bonds are broken, which releases energy in order to power work that the cell needs to get done. Okay, so here's our two laws of thermodynamics. The first law of thermodynamics says that energy cannot be created nor destroyed. And so you'll need to be very familiar with the laws of thermodynamics. You'll need to know them by name, like what is the first law, what is the second law, and you'll need to know those definitions. So energy cannot be created nor destroyed. That means that whatever energy that is inside of the universe is going to be what's in the universe. The universe, although scientists agree that the universe is expanding, but the energy that is here is what we have. We can't add to the energy that's already existing in the universe. On the other hand, the second law of thermodynamics says that energy is going to be transformed. And so, from one type of energy to another type of energy. And so, we'll see that although we can't create any new energy, we can change the formation of that energy that already exists here in our universe. So, a classic example would be this block of ice. This block of ice is very uniform. It has water molecules that are lined up nice and neatly. And then it can change forms, and we can go from what's block of ice into this puddle of water. And so changing from the block of ice to the puddle of water is going to change the arrangement of those water molecules. And so we go from having a nice, even arrangement of those water molecules, and we change over to a very disorderly system in which the water molecules are moving very quickly. They're all over the place. They're doing their own thing. And so this change in um, the formation of the block of ice to the puddle of water is what we call a change in entropy. And entropy is the measure of disorder in the system. And so when we go from a very orderly system to a very disorderly system, we say that the amount of entropy has increased. And so our energy that is used in the universe is associated with, is the system orderly? Is the system disorderly? And we'll talk about as we go through this how making a very orderly system takes a lot of energy to arrange those molecules, as opposed to just letting everything be all loosey goosey, like with the water in the puddle. The change in energy is going from a very orderly system to a very disorderly system, which is associated with energy being released, as opposed to going from an area where there's a very disorderly system to a very orderly system where you have to put in energy in order to achieve that system. Okay, so here's the two laws of thermodynamics as real life examples. And so in this case, we're looking at a plant cell that's absorbing light energy, releasing some of that energy in the form of heat. We'll talk about that heat a little later. That's enthalpy, unusable energy to do work. And then the sunlight is going to activate those systems inside the plant cell. So the plant cell can take on carbon dioxide, it can take water from the environment and use this light energy in order to create chemical energy. It's going to store that energy in chemical bonds, and then the cell can store that energy in the form of potential energy for when the plant cell needs to access that energy later on. So this is a classic example of a potential energy where the plant cells are taking energy from the environment, converting that energy, because remember, energy can't be created or destroyed, it's just going to be transformed. And so then the plant cell is going to store that energy for a later date. Here's the example, the opposite example of our laws of thermodynamics. Here we have an animal cell. Most likely, this is a muscle cell, and so humans, animals are able to eat food from their environment, whether that's plant food or another animal source, and they're going to be able to take that food source. We're going to use carbohydrates as the classic, easy example to follow. And so this animal cell is going to be able to consume the carbohydrates and then convert that carbohydrate into various components, smaller components, and release the energy that's stored in those chemical bonds. As we saw in the previous slide, those chemical bonds were created in photosynthesis. And then the animal cell, in this case the muscle cell specifically, will be able to convert that stored energy into kinetic energy in order for muscle contraction to take place. And again, we can see here that one of the byproducts of doing work is heat. Heat is a form of enthalpy in our energy equation that we'll get to a little later. But this heat can't be used to do any work. It's just a byproduct, a wasted byproduct. But we'll see the laws of thermodynamics working here again, where the energy is being converted. We're not talking about generating new energy, but this converted energy is able to then do some work. So you can see here with the purple ball on the left side, I have it labeled as highly ordered. And so when you have a scenario where there's a lot of little pieces that are coming together to make a uniformed thing, we consider that to be highly ordered because all the little pieces have to be in a very specific place. And it takes a lot of energy. If you've ever done a jigsaw puzzle, you'll know that it takes a lot of energy and effort to put all the pieces together so that it creates one unified thing. We see the arrow moving to the right as an increase in entropy because we're going from a very orderly system to a very disorderly system. And so now you have all the little pieces smashed and they're everywhere, and there's no uniform picture, there's no uniform thing. And so we say that the amount of disorder has increased. And so whenever you have disorder increasing, you have a positive amount of entropy. Again, we're going back to our ice cube to a puddle of water, and you'll see that when we have a very orderly system with the ice cube, the amount of entropy is very low. But as we move from the ice cube to the puddle of water, the amount of entropy is at its maximum because the amount of disorder is very high. So when we're looking at cells and entropy, we have glucose molecule C6H12O6. You'll be very familiar with that chemical formula. We're going to use it over the next two chapters. And so we have glucose, which is what the photosynthesis is going to generate. And then you can see that all the energy is going to be stored in those chemical bonds. It's very organized. We have a low amount of entropy. It is a lot of stored energy in those chemical bonds, a lot of potential energy. But as we move from this glucose to the right side, where we have water and carbon dioxide, which are the byproducts of cellular respiration, you can see that it's a very disorderly system compared to glucose. You have water molecules, you have carbon dioxide, a lot of those bonds are broken, and so you're moving from a very orderly system to a very disorderly system. You're moving from an area of low entropy to an area of high entropy. And this is what takes place between photosynthesis on the left versus cellular respiration byproducts on the right. Here we're looking at a plasma membrane in which we have a transmembrane protein in orange. And this transmembrane protein is going to allow these hydrogens, these ions, we call them just protons because they've lost their only electron and the only thing remaining is the proton. And so we refer to these H plus ions as just protons. And so these protons can't travel across the membrane by themselves. Remember, if it's charged, it's large, those things are not going to be able to move across the membrane by themselves. They're going to need assistance. And so here our assistance is going to come in the form of a transmembrane protein, this channel protein. And so the proteins are going to need to move from an area of high concentration, from an area of high concentration to an area of low concentration. And so the molecules are going to travel through the transmembrane protein to the what appears to be outside of the cell. And so we go from a very orderly system where all the protons are on one side of the membrane, on the inside of the cell, where now we have a equal amount of distributed protons on the inside versus outside. And so you're moving to an area of very less organized, more disorder, and so our entropy has increased. So if you've ever had to clean your bedroom, or if you've ever watched Friends, you may remember this episode. Ross was dating this girl who had a crazy, dirty apartment. And so, again, this is just another silly example of uh, less entropy and more entropy. The top picture has a very clean, you can even imagine that it smells like pine sol. And so this apartment looks like it's takes a lot of effort to maintain this very clean and tidy environment, as opposed to, you know, just coming into your house, kicking off your shoes, throwing your clothes every which way, never taking out the trash. And so it doesn't take any effort to make a mess. And so less entropy when everything is nice and tidy and clean, more entropy when things are just kind of chaotic and all over the place. Okay, so you need to know this equation about total energy. Total energy equals unusable energy plus usable energy. And so we have energy that can be used to do work. That's what we call usable energy. Unusable energy cannot be used to do some work. And so I talked a little bit about heat. Heat is a byproduct of um, some of these chemical reactions, and that heat can't do any work for us. It's just lost to the environment, and so we consider that unusable energy. The usable part of the energy is what's going to power the chemical reactions in order to complete do the cell being able to do some work. So here's the chemical equation, the basic equation. I don't want you to memorize this one, but I like the slide because it has outlined the variables, and you do need to know these variables. I will ask you specifically what is H, what is G, and so forth. And so H represents enthalpy, that's the total energy that's related in the system. G is going to be what we use as Gibbs free energy. This is the amount of energy that can be used to actually do the work. S represents entropy, which we've talked about so far. Entropy is also another form of unusable energy. And then T is the temperature of the system, but we don't use Celsius and Fahrenheit. We use the variable K for Kelvin. Lord Kelvin is the absolute temperature scale. I should also mention here that when we're talking about enthalpy, um, that's also related to the heat that the system is generating. Okay, this is the equation that I want you to know. So this triangle symbol here in front of the G and in front of the H, that triangle means, that's a Greek letter for delta, and delta means the change of. So we're talking about the change of Gibbs free energy equals the change of enthalpy, which is the total energy of the system, minus the temperature on the Kelvin scale, times the change in free energy, uh, the change in entropy, excuse me. And so this equation I want you to memorize because of the difference. I want you to know what happens on one side of the equation versus the other. We can have delta G on one side and delta S on the other side. And so I want you to memorize this equation, which tells us about the total energy in relationship to what's being used and what's available. So this term spontaneous reaction, whenever you see the term spontaneous, I want you to think of a reaction that does not require any energy input. And so spontaneous reactions are going to occur without any input of additional energy. We're not talking about something that's happening very fast. we're talking about something that just can take place on its own because it doesn't require any energy input. And so spontaneous reactions, they're not necessarily fast, but the spontaneous reactions are going to have um, a negative free energy. There's going to be some energy left over, because we're not inputting any energy into the system. So our delta G is going to be negative. We can also associate spontaneous reactions with being exergonic. We've used this term before, EX is prefix. Remember that whenever you see this prefix EX, we're talking about leaving or going out. So here we're talking about energy that is being released. And so whenever we define exergonic chemical reactions, they're always going to be spontaneous. That means they're always going to have a negative delta G. They're going to be spontaneous. They're going to be involved in breaking bonds. Whenever we see breaking bonds, remember that we're talking about hydrolysis reactions. And then here's another term that we're going to use to define these types of chemical reactions. This type of metabolism, which is a sum of chemical reactions, is called catabolic. And so this means that there's going to be a breakdown of bonds, energy is going to be released, and this is defining a collection of chemical reactions in which is spontaneous and exergonic. So catabolic. The way that you can remember catabolic is think of the term catastrophe. And so whenever we have a hurricane that comes to the shore, we can consider that to be catastrophic because the hurricane is going to break up the houses and throw cars and cause a lot of destruction at the coast. And so this is catabolic. There's going to be a lot of breaking down. Energy is going to be released. On the other side, remember we're talking about two sides of the same coin. Well, now we're talking about non-spontaneous chemical reactions. Non-spontaneous chemical reactions are also defined as endergonic. In this scenario, we have a positive delta G. That means we're putting in energy into the system, and so the amount of free energy is going to be positive because we're making an investment. This requires making or building bonds, so we're talking about synthesis reactions, for example, dehydration synthesis reactions, and then we can use this term to describe the metabolism or the metabolic processes as anabolic. You may have heard of the term anabolic steroids, and so anabolic steroids are going to help bodybuilders or athletes build muscle, recover very quickly. And so we're talking about building. And so anabolic is a type of metabolism in which we're building. And so these are the collective terms in which we can use to describe these types of chemical reactions. We can also associate non-spontaneous, endergonic, positive delta G, dehydration synthesis reactions, anabolic metabolism with potential energy because we're storing energy, we're building bonds. And so those terms that we've used previously all go together to describe these types of chemical reactions, the same types of chemical reactions. So there are three types of work you need to be familiar with. There's chemical work. This is work that's going to be associated with the energy in macromolecules, the anabolism or anabolic metabolism. And so we'll talk about this chemical work in photosynthesis and cellular respiration. We've talked about transport, how molecules need to move across the membrane. So, for example, an active transport requires energy. We've talked about bulk transport that requires energy. For example, um, phagocytosis, pinocytosis, right? Those are types of bulk movement of molecules across the membrane. And so endocytosis, exocytosis, those major categories of transporting molecules across the membrane is considered transport work. And then finally, mechanical work. This is the type of work in which we are using our muscle cells, or we're using cilia or flagella. Remember those features of the cell in order to either move, locomotion. The cilia are going to be involved in absorbing nutrients, for example, in the intestines, and so these are associated with moving and doing some type of physical work, and so that's falls under the category of mechanical work. So you need to be familiar with the three types of work that the cells can do. All three types of this work is considered going to be powered by ATP. So going back to spontaneous reactions again are examples of hydrolysis reactions. Remember that our delta G is going to be negative. So here's a real life example. Specifically for ATP, you can see that the delta G is negative 7.3 kilocalories per mole. That's the unit of energy in this particular case, and so the delta G here is negative because energy is being released, because we're talking about breaking bonds of the ATP through the hydrolysis reactions. And so these types of reactions favor the products. The products is what we're after. Energy is going to be liberated from the stored energy of the chemical bonds, and now the energy is released because we're breaking, we're destroying. Falls under catabolic. Remember that term associated with catastrophe or destruction. And so the ATP is being broken down. Remember, ATP is a type of nucleotide. We have our phosphate, or in this case, three phosphates. We have our ribose sugar, that's the blue here, and then we have the nitrogenous base. Because our nitrogenous base is adenine, the A, that's why it's called ATP, adenosine triphosphate. And so this chemical molecules we're going to be very familiar with, because we're going to work on building it in photosynthesis, and we're going to be working on breaking it down in cellular respiration. So there is a huge cycle. Energy is the circle of life, so to speak. And so at the top of the circle, we have ATP fully formed. We have three phosphate molecules that are attached to this nucleotide. And then moving to the right, you can see that is highlighting exergonic reactions, because now we're talking about breaking that phosphate bond in order to release energy. And so we break the bonds. Now we have ADP, adenosine diphosphate. Remember, di means two. And so we have a free-floating phosphate or an inorganic phosphate. And then we have this ADP. And so it has less stored energy because we've broken this bond and released some of the energy. As we move around, going towards the left, in order to restore ADP back to ATP, we have to invest energy. And so this energy investment is endergonic, because we're storing energy in the bonds, in those phosphate bonds, and that requires an energy input. So this is going to be endergonic. It is anabolic. It is non-spontaneous because it takes some energy to invest. So back to our hydrolysis of ATP, you can see that the energy associated with breaking a phosphate bond, releasing the energy, negative 7.3 kilocalories per mole. So this is hydrolysis, a spontaneous reaction, energy is being liberated. Remember that we can go from ATP, which has three phosphates attached, to two phosphates attached, ADP, to AMP, which only has one phosphate attached. And so you'll see these versions of ATP. You should relate that back to how much energy is stored versus how much energy has already been spent. And so ATP has the maximum amount of stored energy versus AMP, which has the least amount of stored energy in its chemical bonds. Remember, this is a hydrolysis reaction, so we're breaking bonds by smashing those bonds with water, and then we release a product at the end. Because this phosphate molecule is not attached to anything that has carbon. We call this free-floating phosphate an inorganic phosphate, which is why you'll see it with a P with a subscripted I. All right, let's get into coupled reactions. Coupled reactions are going to be reactions where you have a scenario of having two chemical reactions that have to go together in order to fuel the reactions, associated reactions. And so ATP is going to be used throughout the process in order to fuel the reactions that require energy. And so we'll see how these couple reactions work together, because what you don't want at the end of a chemical reaction is for the cell to not have energy left over. The system always wants to have energy left over. Just like you want to have money left over in your checking account, you don't want to have to go to work in order to get back to a zero balance. The same thing with your chemical reactions in the cell. The cell wants to couple reactions that cost energy with chemical reactions that are associated with having extra energy left over, and so these coupled reactions will go together so that the cell overall has a negative delta G. Okay, so here's an example of a coupled reaction that we're going to see in the first steps of cellular respiration. In this scenario, we have glucose plus phosphate. This is a phosphorylation reaction because we're adding phosphate to the glucose. That's going to result in glucose, phosphate, with water left over. And so because we have this water left over, we know we're talking about a dehydration synthesis reaction. We're adding a phosphate to the glucose, so we're building bonds. And so this is going to have an energy requirement. It's non-spontaneous. Because we're building, this is considered anabolic. And so you can see here that the delta G has a positive. Delta G is plus 3.3 kilocalories per mole, and you can see that we've also identified this reaction as being endergonic because energy is being stored in the bonds. This is going to be coupled with a reaction that is going to have all the opposite characteristics. So we're going to couple this reaction, this glucose phosphate yielding glucose phosphate plus water. We're going to couple that with ATP plus water. So we know off the top we're talking about a hydrolysis reaction because water is being added to the system. That's going to result in ADP, which has two phosphates attached, and an inorganic phosphate that's left over. And so because we're talking about smashing bonds, releasing energy, we can see that our delta G is going to be negative. And so in this case, it's negative 7.3 kilocalories per mole because we're releasing energy. This is an exergonic reaction. And so it's spontaneous because we don't have an energy input or releasing energy. It is considered catabolic. So these two coupled reactions, one that's costing you energy, is going to be coupled with a reaction that is going to have a excess of energy left over. And so we put these two, energy requirement versus energy left over, together, and then you can see that if we add up those two numbers, we have a negative 4 kilocalories per mole total energy that's remaining. And like I said before, you want to have a negative delta G left over. That means we don't have a energy deficit. We have energy that's left over. It's overall exergonic, which is considered energetically favorable for the cell. And so you should be familiar with these types of coupled reaction. We're going to see examples of them over the next two chapters, in which something's going to cost, and then we're going to couple that with something that's going to have energy left over for a total chemical reaction that's going to have energy left over, because that's what's energetically favorable. The cell is happy when we have energy left over and there's no energy deficit. Now let's kind of switch gears a little bit for enzymes. Enzymes are going to be made up of our macromolecules, proteins. Those proteins are going to be involved in chemical reactions, but they're not going to be consumed during those chemical reactions. They're going to help speed things up, but they're not going to be consumed. And so these are what we call catalysts. You're generally going to see enzymes with ASE as a suffix. Anytime you see ASE at the end of a word, know that we're talking about an enzyme. Enzymes are made up of proteins. These particular proteins are going to act as catalysts. Catalysts speed up chemical reactions, but they are not consumed during the chemical reaction. So whatever catalyst you start off with is going to be the catalyst that's going to be left over at the end of a chemical reaction. We also see this term you may come across in your reading. It's called a ribozyme. Remember, RNA it's similar to DNA, but RNA can also act as a catalyst. And so whenever we have RNA molecule that acts as a catalyst, we call this a ribozyme. It is not going to be a part of the category of enzymes, but it's going to do enzyme-like work. And I had, this is an old slide um, where I used to have a comprehensive final exam. We don't have that anymore, so ignore this notation about this being on your final exam. You will see reference to it on exam 3, however. So in a metabolic pathway, this is going to be a series of chemical reactions that take place. And remember, metabolism is referring to the sum of these chemical reactions. And so A, B, C, and D are identified as individual chemical reactions. And then we see the E1, E2, E3. These are going to be the enzymes necessary to facilitate those chemical reactions. And so in order to go from A to B, we're going to need an enzyme. But now B is different from A, but we want to go from B to C. And so B is going to need another enzyme in order to convert B to C. And then, just like before, C is different from B, but now we need C to go towards D. We can't use E2 anymore. We're going to need a new enzyme to do this conversion from C to D. And so that new enzyme comes in the form of the third enzyme, E3. So the reactants are going to be what the enzymes are going to be using in order to do the conversion. And so the thing that's being worked upon is going to be the reactants, also called the substrates. The product is what you end up with, but those products could also then become a substrate themselves. And so A starts off as a reactant or the substrate, which gets converted into B, which is the product, but then that B is going to then be the new reactant or substrate for enzyme E2, and so forth. So how many people have to use an alarm to get out of bed in the morning? Do you have to set one alarm, two alarms, three alarms, put alarm clocks all over your bedroom? Well, the amount of energy it takes for you to get out of bed in the morning is associated with your activation energy, how much it takes you to get up and going in the morning. Your chemical reactions also have what is called an activation energy. How much energy is it taking to get this reaction from the reactant side to the product side? And so this initial input of energy, just like that initial energy you have to use in order to get out of the bed to get the reaction started or get your day started, this is called the energy of activation. What's happening on a molecular level is that you have these molecules that have to get close enough to cause the bonds associated with that molecule to be rearranged. And so this is going to be something that could potentially take a very long time for it to go forward in order to overcome this energy of activation. But once you've reached this transition state, which is right there at the peak of our heel, um, that our little man is pushing the boulder over, right there at the top of the heel, we call this the transition state. So the bonds of the molecule that's about to change are going to be stretched to their limit. And then once we get up and over the hill to the product side, those bonds are going to be rearranged. And so we have this little man that's pushing this boulder up and over the hill. And so to get this boulder over the hill, to overcome that activation energy in order to get to the product side, it requires a lot of energy. And so this hill, this barrier that he has to overcome is what we note as the activation energy. And so all chemical equations, all chemical reactions have this barrier, this energy barrier that must be overcome. So overcoming this activation energy, in order to do that, we can do that two ways. We can introduce heat to the system, and so this heat is going to allow these molecules to get closer to each other in order to facilitate this chemical reaction. So heat can be a factor, or we can use an enzymes. And in most biological systems, we don't want to increase the amount of heat. The amount of heat is also associated with enzymes being degraded. And so we don't want to create a scenario in which heat can get out of control. And so the body in particular is going to prefer to use enzymes to overcome this activation energy. And so you can kind of think of it as cutting this hill in half. So, or imagine that the hill wasn't there at all, and this man could just push the boulder straight off the cliff instead of having to go up and over, way over the cliff. And so overcoming this activation energy, we're going to talk specifically about using enzymes. So in this slide, we can see that there's an enzyme. This enzyme has a place for the substrate, and in this case, the substrate is the ATP and the glucose that have to come together. And what's going to happen, and we'll see this in the next few chapters, is that a phosphate molecule is going to be broken off of the ATP, and then that phosphate is then going to be added to glucose. This is that coupled reaction we saw in the previous slides. And so the substrates have to come close together in order to facilitate this chemical reaction. And by bringing these molecules together, that's lowering the activation energy that's required to move these chemical equation from the reactants to the products. So the enzyme is going to facilitate this reaction without the use of adding additional heat to get these molecules to come close together. Otherwise, it could take minutes, hours, years, tens of thousands of years for this chemical reaction to move forward on its own. So we start off on the left-hand side with the reactants, and you can see that we have a we can call this starting place 0. And so the free energy above or below this line we can consider positive or negative. The red line is the use of the chemical reaction without an enzyme, and the blue line is demonstrating what would happen with our chemical reaction with the use of the enzyme. And you can see this hill that we have to overcome. Remember, I told you that was the activation energy. This activation energy is dramatically reduced, maybe even by half, by using the enzyme. And so we have our reactants, we come up and over the hill, whether that's with or without an enzyme. And where you start to where you have to go, this is the energy of activation. So you see the energy of activation without the enzyme is much higher because the hill to overcome, this energy barrier to overcome is much higher. And then the blue line is showing us from the reactants to the top of that hill is much lower, and this is the activation of energy with the enzyme. And so if we move past this energy barrier, the transition state, down to the product side, it shows you that no matter which route we took with an enzyme or without an enzyme, we get to the product side and we end up at the same location either way. So reactants to products, eventually we'll get there. How we get there, how fast we get there is the difference between using the enzyme and not using the enzyme. And so you can see with this chemical reaction that the products is much lower than where we started. So remember, where we started with reactants, we can consider that a 0 point. And so because we're much lower than that reactants starting point, then we can say the change in free energy in this scenario is negative because it's below the 0 line. We did have to input energy, and so above that line would have a positive delta G. So this means that this is a anabolic reaction. It's non-spontaneous because it requires an energy input. But overall, the reaction is exergonic, it's catabolic. We have free energy left over, so it's spontaneous. So because this is a coupled reaction, we can input some energy, but overall we have a exergonic reaction, catabolic reaction. So again, using the enzyme is going to help us lower the activation energy because we're going to be able to position these reactants to facilitate a bonding. We're going to bring these molecules close together to encourage a chemical reaction. And that's the basis of using an enzyme, is because we can position these reactants in such a way, these substrates in such a way that we can facilitate a chemical reaction without having to wait to overcome that energy of activation. So here again is our enzyme. You need to be familiar with these terms because we're going to talk about them in the photosynthesis chapter and in the cellular respiration chapter. We have an enzyme. We have the ATP that needs to be bound to the enzyme, and this is a substrate. We also have glucose in this scenario that has to also be bound to the enzyme. And so both of these are substrates because they're binding to the enzyme. So you should also know that where the substrates are binding on the enzyme is also called the active site. This is where the chemical reaction is going to take place. So once you have all the components together, the enzyme, the substrates bound to the active site, we enter a new stage, which is called the enzyme substrate complex. And scientists like to use this term complex when they're describing how multiple things come together. And so because we have these two types of substrates coming together to bind with the enzyme, this is called the enzyme substrate complex. We can see with the enzyme substrate complex, there's a chemical reaction that's taking place there at the active site. This is called a intermediate because we're not quite to our final product, but we're still within the enzyme substrate complex. And then finally, at the end of the chemical reaction, we release the products. And so because we took one of those phosphates and added them to the glucose, we've lost a phosphate on the ATP. So now that is known as a DP because it now only has two phosphates attached. The glucose now has a phosphate, so it's forming a glucose phosphate molecule. They're being released from the enzyme, and you can see that the enzyme from the beginning through the enzyme substrate complex modification to the very end of this, we have a product. The enzyme has stayed the same. It has not been consumed in the chemical reaction, which is characteristic of an enzyme. So the substrates bind to the active site of our enzyme, and this binding is highly specific. And so the active site has a high affinity for the substrates that binds to that area. And so there's two ways that you can have binding of the substrate to the active site. You can have what is called the lock and key. For example, if you had a key to your car, to your house, or your apartment, your key only opens the door that the key is associated with. And so there's one enzyme, one substrate. You can also have a scenario where the substrate binds to the enzyme and it causes the enzyme to change shape to accommodate the substrate. So we call that an induced fit. You can consider that like having a master key for a building. One key would unlock multiple doors. It would be similar because the key would make the door lock somewhat change shape on the interior mechanism that would accommodate that master key, change where the locations of the lock pins are in order for the door to be opened. And so the induced fit is like this master key. It's going to bind to the substrate. The substrate is going to bind to the enzyme, and the enzyme is going to change shape to accommodate the substrate that is bound. And we can see here the substrate binding to that active site. You can see the active site does not have a specific shape that the substrate would accommodate, but once the binding takes place, the enzyme is going to change its overall shape. There's a conformational change that will take place in order to accommodate the substrate at the active site. So remember I told you that whenever you see the name um, of one of these chemicals, for example, lipid, urea, maltose, RNA, lactose, these are all substrates. And whenever you see a name that has ASE on the end of it, we're talking about the enzyme. And generally speaking, the enzyme name is the name of the substrate that's going to be accommodated. So a lipase is going to accommodate lipids. Urase is going to accommodate urea. Maltase accommodates maltose and so forth. And so the substrate's name is usually associated with the enzyme name. Not always, but generally speaking, especially for the introductory material that you guys will see in this class, it would generally be very obvious what the substrate is as opposed to the, or in combination with the enzyme name. Later on, we're going to talk about coenzymes. I put this in here because we will see it in photosynthesis and cellular respiration. That chapter number has changed, but you will see reference to a coenzyme. These are organic molecules going to participate in chemical reactions, just like enzymes are going to be left unchanged. And so you'll see reference to coenzymes in the upcoming chapters. This graph, you need to be able to read graphs similar to this. I told you before that enzymes are susceptible to their environment, and so enzymes have to have a optimal pH. Enzymes have to have an optimal temperature range in which they're able to function. And you can see on the y-axis we have the rate of chemical reactions, and you can see you go from 0 to this arbitrary high. And so at the top of the red graph here, we can see this is where the enzyme is optimal. And so the temperature slightly above and slightly below is where the enzyme is not going to function very well, but you can see there's a range in which the enzyme works best. When we're talking about temperature, temperature is a little bit more forgiving on the cold side, and so the enzyme doesn't drop dramatically to 0 for the rate of chemical reactions. It kind of gradually declines. There's a tail on the left side of the graph showing you that the enzyme can work, it's just not going to be working as effectively as you approach 0 degrees Celsius. But on the other end of the chart, you can see that just outside of 40 degrees, the enzyme rate of reaction drops dramatically to 0. So this is a scenario in which you can have some real life application. People that fall off boats or get trapped in very cold water, a lot of times they can be recovered and they can be resuscitated and make a full recovery if they fall off into really cold water, as opposed to a scenario where an individual is maybe trapped in a hot car or they get a very high fever. It's unlikely that you would recover from having a temperature that is too high for too long, as opposed to having a good prognosis of coming back from being in a scenario where there's hypothermia. And so temperature on the cold side, again, is somewhat forgiving for enzyme reaction rates, but it's not very forgiving when the temperature is too high. Either way, if we're talking about having the temperature too high or too low, we call this denatured. I've used this term previously. I'll show you in the next slide this term again, denatured. And so above and below the optimal range, the enzyme is not going to function very properly. You can see a pH graph that could look very similar to this. There's going to be a range where the enzyme is going to be optimal, and then there's going to be a range in which the pH is too high or too low for the enzyme to function, and the rate of reactions will plummet to 0. Here's that term here in bold, denatured, again at high temperature or low temperature. At high or too low pH, the enzyme is not going to function properly. It's going to unfold. Unfolded protein is not functional. Unfolded protein, unfolded enzymes, to be more specific, is not functional unless it's folded. And so temperature and pH are very important when we're talking about the effectiveness of enzymes. Here on the left, the picture here is showing you another temperature graph. And again, we can replace that temperature with pH to show a similar graph for the function of the enzymes. And so body temperature is very important. You can think of animals that are dependent on the sun. They have limited rates of chemical reactions when their body temperatures are either above or below their optimal rate. Body temperatures of endothermic animals are a little bit more forgiving because of the way their internal system is maintained. Okay, we've talked about pH over the last few slides. Again, there's optimal pH, there's optimal temperature. Outside of this range, the enzymes are not going to function very properly. And here's those graphs. You can see they're very similar to the temperature graphs. We have some enzymes that are like to be in very acidic conditions, and that's 0 to 6 would be considered acidic. And so pepsin is a enzyme that's found in the stomach, and you see its ideal temperature is about 2. Above or below that, the enzyme reaction rate is going to plummet dramatically towards 0. Trypsin is another enzyme. It has a slightly higher pH, so the optimal pH for trypsin is 8, but again, above and below that pH range, we can see that the enzyme activity plummets to 0. So there's an optimal pH. Not all enzymes have the same optimal range, and so you have to keep that in mind. You will see graphs similar to this on your next exam. So enzymes may also be involved in activating molecules so that they can be their active versions. And so we're going to see a lot of use of a particular enzyme called a kinase. It's a family of enzymes, and the jobs of this family of enzyme is to phosphorylate things. And so phosphorylation just means adding a phosphate to a molecule. And so in this case, we have an inactive protein that a kinase is going to act upon that inactive protein. And then we're going to end up with a product that has 2 phosphates that are attached, which will then generate an active protein. And so in this scenario, this modification may even take place in the area of the cell, somewhat like in the Golgi apparatus, where modification of proteins would take place. The activation or deactivation of proteins can be associated with genes being turned on or turned off. We're going to talk about that towards the end of the semester. But activating these proteins is necessary to go from an inactive version, an unusable version, to a version that will able to be involved in some metabolic pathway. Sometimes we can inhibit an enzyme. Inhibition means to turn it off or reduces um, activity rate. And so inhibition occurs when the substrate cannot bind to the active site of an enzyme. And so if you block this in some form, then you can prevent the enzyme from working on that substrate. This can have positive and negative consequences. And so we'll talk about some of those reasons why you would have inhibition coming up. One of the types of inhibition would be to block further production of the product. Maybe you don't need this product. Maybe this product would promote cellular death, or this product might allow for a cell to move into the next phase of cellular division, even if it's damaged and could be considered cancerous. And so sometimes you want to block the way that enzymes would normally work in order to regulate a system. Okay, so the more complex enzyme inhibitions, we can see this be an example of a product binding to a site other than the active site, which changes the shape of the active site, which will then prevent the enzyme from binding to the substrate. And so this is a type of inhibition that's a multi-step process. And so we can see examples of this being with cyanide. Cyanide inhibits enzymes from binding in a pathway of allowing the red blood cells to bind to oxygen. We can see in penicillin blocks the active site of bacteria enzymes. So it's a poison to bacteria in order to promote cell death, which in case of bacterial infections would be a positive. And so there can be a complexity of enzyme inhibition, which can be negative or could be positive for the individual. So enzyme inhibition, here's an example of positive and negative feedback. In the top half, we have an active pathway where a reactant is going to bind with the enzyme, and then it's going to produce eventually some product F, okay? And so the reactant and the E enzyme has to come together in order to facilitate these chemical reactions, which will eventually end up with a product. And then that product would then bind to the enzyme, change the shape of the active site in order to prevent the reactant to bind to it. And so this is what is considered a negative feedback, because the product that is generated is then going to turn around and turn the system off. We could also have a positive feedback mechanism where the end product would promote additional reactants to bind to the enzyme to facilitate more products. But in this case, we have a negative uh, feedback because the product that is produced by the first chemical reaction therefore turns around and turns the system off and prevents the reactants from binding to the enzyme. So here's an overview of the things that we've talked about so far. We talked about metabolism in general. We talked about anabolic versus catabolic. In the first part, we talked about chemical reactions in a metabolic pathway. We talked about using enzymes to facilitate going from the substrate to the products. We talked about how if that pathway is destructive, if we're talking about breaking things down in order to release energy in an exergonic formation, using hydrolysis reactions, having a negative delta G, these are chemically, genetically favorable. These are also, we could use the term spontaneous reactions that could take place. And so that's half of our double-sided coin. The other half would be anabolic pathways. These are building. They're going to promote synthesis, for example, dehydration synthesis. We're investing energy, so it's endergonic. We have to couple these reactions with exergonic reactions. Remember, the cell doesn't always like to be investing, and so it's going to couple these reactions that are costing energy with reactions that's going to have energy left over. These reactions we can also consider dehydration synthesis reactions. We have a positive delta G, and these are overall energetically unfavorable because it's costing the cell energy. And so if you can kind of put together all of the things that we've talked about so far together in two major categories, even back to potential versus kinetic energy, and just kind of understand why we put the terms on one side versus the other, then I think this chapter makes much more sense. It's going to be a great foundation for us as we move on to the photosynthesis chapter. The last thing that we'll cover is the idea of substrates, intermediates, and then the final product. So in this slide, we have a picture of glucose. The glucose has these hydroxide groups, these OH groups that are attached. And so in what we'll see in the next few slides, we're going to replace each of those OH groups with a phosphate. So everywhere you see OH, we're going to replace those OH, those hydroxyl groups, with a phosphate. An enzyme is going to do this work, but every time we change the molecule, we change the substrate, which means we need a new enzyme. So here we go. On the left, we have the initial substrate. This is glucose. We're going to use enzyme 1 to convert that glucose into a phosphorylated molecule. We've added a phosphate to carbon 1, 2, 3. Okay, remember we start counting from the right of the oxygen. So everywhere there's a corner, there's a carbon that's associated there, and we start counting. So at carbon 1, 2, 3, we have replaced that hydroxide group with a phosphate. And so we have a new product. It's not our final products. We call this our intermediate. So our intermediate product has to then be a substrate, and so it's going to be a substrate to a new enzyme because enzyme 1 can't work on this substrate. It's, remember, glucose would be specific to the enzyme 1, and we don't have just straight glucose anymore. We have a phosphorylated glucose, and so we need a new enzyme to act upon our new substrate, which is intermediate 1. So we're going to phosphorylate this at carbon 1, and so carbon 1 is going to be phosphorylated by enzyme 2. And remember, the types of enzymes that do this phosphorylation are called kinases. So we can move from our intermediate 2 into our final product. In the next chapters, cellular respiration is a catabolic pathway. We're talking about breaking things down. We're talking about releasing energy. In the cellular respiration chapter, we're going to see energy stored in the form of various intermediates such as ATP, which is not one of our final products. We're going to see NADH, FADH2, and so storing energy is going to be something that's going to happen to capture electrons temporarily and then get those electrons back in order to make lots and lots of ATP at the final product of cellular respiration. And so we'll explore these catabolic reactions in chapter 8. There's a few ways we can make ATP. We can do that via substrate level phosphorylation. That means we're going to use an enzyme to directly add phosphates to an adenosine molecule in order to generate ATP, or we can use chemiosmosis. And so this is going to be how we create an electrochemical gradient. We talked about that in the last chapter. So we can create this electrochemical gradient. We can use the proton motive force in order to facilitate the chemical reaction to convert ADP or AMP to ATP. And so we'll talk about this process in chapter 8 and in chapter 7. So we'll come back to it. I'm just giving you a preview of things to come. So if the terms don't make sense now, don't worry. We're going to have time to explore them in the upcoming chapters. Okay, one of the last things that we're going to talk about for this chapter is redox reactions. I told you about these coupled reactions, but now let's look at half reactions, where where we're showing you how we can follow different portions of a chemical reaction and see how it's changing an overall chemical reaction. So some things in the chemical reaction are going to gain electrons, some of them are going to lose electrons. And so whenever you see a species or a component of a chemical reaction that loses an electrons, that means electrons are going to be on the right side of the chemical equation. We call that oxidation. Do not get it confused when you see this term oxidation. We're not talking about oxygen. We're talking about the loss of electrons. So this species, this element, this component is going to become more positive because it has lost electrons. On the other hand, we're talking about reduction. And so part of the overall chemical reaction, we're going to have something in the reaction that's going to be gaining electrons. And in this scenario, you will see electrons being added on the left-hand side of the chemical equation. So in this example that I'm showing you here, we have hydrogen. Hydrogen is going to be losing electrons. You can see the 2E on the right side of the chemical equation. And then we have fluorine, which is going to be gaining electrons. And you can see we're adding 2 electrons on the left side. And then at the end, the fluorine has an overall negative charge. And so the fluorine in this case is being reduced. The thing that's being reduced is its overall charge because it has gained electrons. So combine these two parts together, oxidation and reduction, we call those redox reactions. And so you're going to see in the next few chapters, you're going to see some things that are going to be gaining electrons. You're going to see some things that are going to be losing electrons, follow the electrons, be able to use these terms oxidation and reduction in order to describe what's happened. Remember the first law of thermodynamics. We talked about it way back in the beginning of this chapter. The first law of thermodynamics says energy can neither be created nor destroyed. So this energy has to go somewhere. If we take away electrons from hydrogen, it doesn't just disappear, it's got to go somewhere. And so that means those two electrons that are released, the fluorine is going to pick them up. This is going to facilitate the overall chemical reaction that takes place. We'll get some practice. We'll spend a lot of time practicing identifying these components in the upcoming chapters. Here's another general example. We have A, we have B. A is going to, A with the electron is on the left side is going to be converted to just A by itself. B on the left side is going to be converted to B with an electron on the right side. And so in this case, A is being oxidized because it's losing an electron. And B in our scenario is gaining an electron, and so it's going to be reduced. And so the thing again that's being reduced is the overall charge. We're going to see energy intermediates in the next few chapters. One of those energy intermediates in cellular respiration, for example, is NADH, nicotinamide adenine dinucleotide. You'll never see that written out ever again. And so NAD becomes NADH. NAD is in its oxidized form. It's missing an electron, but it can gain electrons, which is going to be some of the intermediates that we're going to see. And so NAD to NADH has become reduced because it picked up electrons along the way. We call these H pluses protons because hydrogen, if you look back at the periodic table, hydrogen has one proton, one electron, and so if it's lost this electron, the only thing that's remaining is the proton. So sometimes you'll see reference in your textbook proton when referring to just the H plus ion. Here's what that NAD looks like, and you can see at the very top in the grayed out box, we have a nitrogen that has a positive charge in the ring structure. And this is where those electrons are going to go. This is where they're going to be stored in this nitrogen on the ring. And so in this case, we have NAD that's being reduced, is gaining electrons, and this is a reversible process. And you can see on the left side we have a oxidized version that is becoming reduced. Moving from left to right, you can see we have a molecule that has a positive charge, and then on the right side, it's neutral. And so in this case, this neutral version is NADH because it has picked up this additional electron, has picked up an additional proton. And so we're going to see NAD, NADH. These are our intermediates that we're going to be playing with in our cellular respiration chapter. Okay, so again, anabolic, we've talked about catabolic, anabolic. Our examples for anabolic is going to be with photosynthesis. These are biosynthetic reactions. That means that the plants are going to be synthesizing this from the energy that is gaining from the sun. And so we're going to take smaller molecules. For example, we're going to take CO2, we're going to take water, we're going to use light energy. We're going to convert those carbon dioxide molecules into sugar. And so we're building macromolecules from smaller molecules. This is anabolic. I like this figure. ADP, I showed it to you a type of this figure before. ADP picks up a phosphate. We have an energy input. That means it's endergonic. That means dehydration synthesis is taking place. And we end up with the ATP molecule with water left over. Now that water can then be used in order to smash those bonds, release energy. And so you'll have an input of energy, dehydration synthesis. We have hydrolysis reaction where the energy is going to be released in an exergonic formation. And so it's just a circle of life with the ATP that we're going to see in photosynthesis and cellular respiration. Okay, our last slide here is just a preview of how we're going to see metabolic pathways regulated. I talked a little bit about it before. Turning genes on and off is a type of gene regulation, metabolic regulation. Cell signaling pathways with hormones can turn systems on and off. That's another type of regulation. And then feedback. We talked about feedback inhibition, that negative feedback, or positive feedback as an example of metabolic regulation. And we'll explore some of these things in more details later on. I mentioned negative feedback, but we also have positive feedback, where the products that are produced can turn the system on. If you think about when a woman starts having contractions, which will eventually lead to the birth of a baby, those contractions are stimulated by hormones, which increases the additional release of hormones and causes for additional contractions to take place in order for the baby to be born. And so that's an example of positive feedback for what you produce turns on the system to cause even more of the products to be generated. So gene regulation, cellular regulation, biochemical regulation are all examples of regulation of metabolic pathways. And again, we'll explore some of those things in more details later on. All right, that's the end of this chapter.